Good afternoon, ladies and gentlemen. My name is Amy Harvey, and I'd like to discuss with you today the possible effects that glyphosate and Cry1 proteins can have on tadpole development. Pesticides play a large role in the agricultural system and is well used amongst them. There are two types of pesticides, herbicides and insecticides, which can be applied both externally or internally by means of genetic modification or sprays. Pesticides can find their way into the ecosystem and can affect aquatic life such as fish, insects, and amphibians. And previous studies have also shown that cry proteins at different levels was found in surface waters of streams and rivers. In our study, we looked into the effect of Roundup and Bt maize. Now, Roundup is only a short topic we speak about, and is an active herbicide with an active ingredient, glyphosate. Genetically modified maize, on the other hand, is the removal or addition of a specific DNA sequence from an organism. The gene in this case is removed from Bacillus thuringiensis, or soil bacterium, and transferred into the maize DNA, hence the name Bt maize. Bacillus thuringiensis has pesticidal traits which protect the maize from different insect activities. Sorry, I must just forget that one. Here is the non-Bt, where there's no pesticidal traits, and you can clearly see there's insect damage, whereas the Bt has none. Now, this gene that is transferred into the maize DNA produces Cry1 proteins, which is toxic to specific insects. The protein we use is Cry1Ab, which is specifically toxic to the African maize stem borer. We chose amphibians as our test organisms because they are good indicators of ecosystem health. They absorb substances because of their permeable skin and can transport nutrients from one ecosystem to another. They are also suitable for monitoring processes due to the abundance of knowledge known on this group, the role in the different trophic levels, their breeding site predictability, as well as their easy accessibility. We made use of two species, Xenopus laevis and Amitophrynus guturalis. The reason why we chose these two species is because when you plot them on a graph, they overlap with the production of the maize. They overlap with the production of maize. In this case, the Xenopus laevis is found all across South Africa and also overlaps with the green area, which is the production of maize. The guturalis is found in the high regions of South Africa and still yet overlaps. Our aims and objectives were to determine the appropriate husbandry practices for Xenopus laevis tadpoles, as well as whether the Cry1 proteins released from the genetically modified maize leaves will have an effect on the tadpoles. We also wanted to determine how glyphosate would have an effect on the development on Amitophrynus guturalis, and whether Cry1Ab proteins are actually released from the leaves and when they will start to degenerate. Our first experiment was to determine which husbandry practices would be best for the Xenopus laevis tadpoles. To do this, we looked at three different factors: the food type, container type, as well as the treatment type. This is how our experimental setup looked like. We had the tanks at the back and trays in the front of each tank with 15 cups in each. 60 tadpoles were placed in the tanks and one individual tadpole in each of the cups. The tadpoles were acquired by induced spawning. This is our experimental setup. We had two different groups, a borehole treatment, and a non-Bt infusion. And in the first two trays and tanks of the borehole and infusion was fed with tetratabamine, and the other was Xenopus food pellets. Throughout the experiment, I'll refer to them as the SAEON pellets. They have the exact same content as the Xenopus pellets. Okay. When you compare the different containers, we found there's a large significance between the two containers. Xenopus laevis are social organisms, and here you can see they thrive in the groups in the tanks where there were 60 tadpoles, and didn't do so well in the cups. When we looked at the treatment, we also found a smaller significance, but yet still a significant difference between the infusion and the infusion and the borehole water. Now, the infusion was non-BT, so we can't say it's BT that's caused the decrease. But further investigation should be done on the contents of the leaves or even the water clarity, which can affect the tadpole growth. When we looked into the food differences, there was no significant difference. However, when we made a pie chart, we saw that those in the non-BT infusions had a high percentage more malformations. And when you look at the food, the tetratabumin had the highest percentage malformations. Now, this is how a healthy tadpole should look. This tadpole was taken from the borehole water fed with the Xenopus pellets. This one is that was housed in the infusion and fed with tetratabumin. Now, we refer to this malformation as scoliosis due to the bend in the spine. When we looked at the mortality, we found that the tadpoles housed in the borehole water and fed with the pellets had the lowest mortality number. However, those that was fed with the tetratabumin, although also housed in borehole water, had the highest mortality. So somewhere along the line, the tetratabumin was not sufficient for them. The two infusions were very close to one another. At the end of this experiment, we found that the borehole water produces a faster growth rate in the tadpoles, or at least a normal growth rate. And the Xenopus pellets, or lucerne pellets, have less scoliosis. And we also found that the Xenopus tadpoles thrive in the tanks. And using this data, we were able to prepare our next experiment. Our second experiment was to find out whether the Cry1Ab released from genetically modified maize leaves will have an effect on the tadpole development. To do this, we looked at 4 different treatments. The first 2 was BT and borehole water and a non-BT, which was used as a control. The last 2 was BT and non-BT isolines. Our experimental setup looked as follows. We had 4 groups of 4 replicates, which were rearranged twice a week to make sure that all the groups were exposed to the same conditions. To make our infusion, we submerged 36 grams of BT and non-BT maize leaves in 10 liters borehole water. 60 tadpoles were housed in each of the tanks and were fed with finely ground lucerne pellets, and their tanks were also cleaned once a week. They were also measured and their developmental stages determined weekly using the NF staging table by Newcomb and Farber. When we looked at the length, we found there is a large significance when comparing that of the borehole water to the non-BT control, as well as the non-BT and BT isolines. Now, what we found interesting was that the 2 non-BTs were significantly different to one another, suggesting that there is a probability that either the maize hybrid has an effect on the tadpoles or the different strains. When we looked at the percentage mortality, a similar result was found, with the borehole water being the most appropriate, with the lowest mortality rate, and the non-BT followed that, and the BT and non-BT isolines were very close to one another again. The developmental stages showed once again the same results, with the borehole water having the highest developmental stage, followed by the non-BT control and the BT and non-BT isolines close to one another. At the end of this experiment, we found that BT is not the main contributor to mortality, and that rather the infusions can have a negative effect. We also believe or hypothesize that there's a probability that the production of the maize leaves, maize hybrids, have different effects on the amphibians, as well as the toxicity can depend on the different strains. We also opened a new question. Is water clarity an important factor to tadpole development, or even the content of the water itself? Our third experiment was to determine whether glyphosate has an effect on Amitriphanus guturalis. To do this, we used 4 different volumes of Roundup. 4 buckets are filled with 5 liters borehole water, and the above-mentioned volumes were added to these buckets. Our experimental setup looked like this. We had 15 different trays, and in each tray 15 cups. These cups were filled with 250 milliliters of the corresponding treatment. 1 tadpole was placed into each cup and had its own oxygen supply. They were fed twice a week with lucerne pellets, as well as their caps cleaned once a week. The developmental stages was also determined weekly using the Gosler staging table. At the end of this experiment, we found that the 3 highest concentrations had 100% mortality before 5 weeks, and the maximum mortality in the borehole water only reached 7 individual deaths at week 3, and that of the non-BT, the lowest concentration, only reached its maximum mortality at week 7. When we looked at the developmental stages, we found that there was no significant differences. However, when we compared the number of individuals that reached metamorphosis, we found that there is a difference. So though they reached the same developmental stage, not all reached at that time metamorphosis. So the borehole water, the tadpoles exposed to the borehole water, had, during the 21-week experiment, 33 individuals that reached metamorphosis, whereas that of the lowest concentration, only 17 reached metamorphosis. At the end of this experiment, we found out that the higher the concentration, the higher the mortality, and the lower the concentration, it can only lead to a reduced developmental stage. We also found that the surfactant, or recent studies have found that the surfactant might enhance the toxicity of the glyphosate, and that should also be taken into account when doing a similar experiment. Our last experiment was to determine whether the Cry1Ab proteins are actually released from the maize leaves and how long it will take for them to degenerate. To do this, we submerged 24 grams of BT maize leaves in 1 liter borehole water and purified water. This was done at 3 different temperatures, 10, 21, and 30 degrees, to account for a night, room, and day temperature. This was repeated again with non-BT maize leaves. The replicates are stacked on top of one another to make sure that the leaves remain submerged during the trial period. Samples were taken at the selected time slots and frozen at negative 81 degrees. Once all the samples were collected, an ELISA was done using Envirologic's QuantiPlate kit for Cry1Ab proteins. We found that the higher the temperature, the higher the concentration Cry1Ab proteins are released, and the lower the temperature, the lower the concentration. This is now for purified water, and we found more or less the same result in the borehole water. However, in the borehole water, there was a lot more fluctuations with the data. So at the end, we found that the low temperatures lead to low concentrations, high temperatures lead to high concentrations. And a reason for this is that high temperatures denatures plant membranes, which allows more proteins or more substances to leach out of the plants, leading to a higher concentration. We also found that during the first 16 and 24 hours, there was very little growth. And if any other experiment should be done, you should focus on times further than or later than 1 day. Yeah, and I've discussed that. Our overall conclusion was that both herbicides and insecticides can have an effect on the BT tadpoles at extreme high concentrations. But, however, the concentrations that we used was environmentally relevant, and therefore we found no prove that it is actually the BT that can have an effect on the development. We also found that according to the studies that we did, that mortality is not the main contributor to mortality. Thank you.